Hi, everyone. Welcome to the next session. I'm excited to have Gianluigi and Maurizio will introduce.
Hello, everyone. Andrea, you give me the okay when I can start?
Yeah, you can start.
Okay, terrific. Good morning, everyone, again. This is Maurizio De Pita speaking. We are now introducing Gianluigi Mongillo from the Institut de la Vision and the CNRS and the Sorbonne University in Paris, France. For those of you that do not know maybe where Paris is, but I doubt it. And we are in the synaptic physiology session, the opening session of the CCNS. And let me introduce briefly who is Gianluigi. So besides being a dear friend and colleague, Gianluigi started doing his physical training in physics in Rome and the University La Sapienza, being one of the students of Daniel Amit. And then he pursued at the same university a PhD degree in neurophysiology. And he pops up every now and then across different geographical areas, including Israel and somewhere in Europe. And now is stable in Paris for the past 10 years. And is, yes, I don't know, you sounds very, you look very disappointed by that. But
no. It's 15 years actually.
15 years, okay. And Gianluigi has developed an expertise over the years in the study of mechanism of synaptic plasticity, and in particular short-term plasticity. In the past, showing important influences of this mechanism in the onset of working memory, and later, I mean, he has spanned many topics in synaptic plasticity, and today he is talking to us about the memory volatility topic that I touched before. Without further ado, Gianluigi, the stage is yours. Please take on.
Thank you, Maurizio, for the nice introduction and for inviting me. Of course, I'm very happy to be here and to be talking about this work that I've been doing together with Jonathan Levenstein at the Hebrew University and Simon Rumpel at Mainz University in Germany. So this is the outline of my talk. I'll start by describing shortly some recent experimental evidence about inhibitory synaptic plasticity. So also inhibitory synapses, they undergo modifications, in particular as a result of some kind of behavioral learning. And I will discuss different approaches or ways to interpret theoretically, to some extent, the significance of this kind of plasticity. In particular, a very influential hypothesis is this homeostatic hypothesis that I will be discussing shortly. And I also want to present a different way to, let's say, theoretically think about inhibitory plasticity, and show that, in fact, if you do this kind of inhibitory plasticity can be naturally understood within a standard context framework, the so-called Hebbian framework, which generically relates long-term plasticity to memory storage, and hence the name storage hypothesis that also I will be detailing. Then I will show by using kind of theoretical and numerical tools that, in fact, inhibitory modifications are suitable for storing memories in a way that is essentially consistent with the experimental data that we have. And more interesting to some extent, I will also show that in some kind of context, actually inhibitory storage can be significantly more effective than excitatory storage. And eventually, at the end of this, I will, summarize a little bit the results and discuss some of the limitation of this work and some of the perspective that this work opens. So let's start with inhibitory synaptic plasticity. So there are essentially, kind of what people mean by plasticity is essentially very ill-defined and mostly depend on the kind of the experimental or the type of experiments you're running. So let's say at one extreme or at some kind of reasonable extreme, there is a microscopic level. So essentially you try to probe synaptic dynamics or synaptic plasticity in this specific case at the single synapse level. So a kind of standard protocol for doing this is the so-called spike time independent plasticity, where essentially you control pre and postsynaptic activity, and then you pair pre and postsynaptic spikes with some specific interval. And this is illustrated here for an inhibitory synapse and for excitatory synapse. So typically you have a pre-post condition or post-pre condition. So this refers to the relative timing of pre and postsynaptic spikes. And in this case, you can see that in fact, when the interval between pre and postsynaptic activity is within 10 milliseconds, positive or negative, you will have a kind of long-term potentiation, so the synaptic efficacy will increase. And if the coincidence is of pre and postsynaptic activity is outside of this window, essentially there will be depression or no change. Here for reference is the same protocol applied to excitatory synapse. And here you can see that the presynaptic postsynaptic will lead to long-term potentiation, and vice versa, this will lead to short-term depression. This is an example, and just to give you a kind of a feeling of the complexity of this phenomenology, this is, maybe at the other extreme. This is by using immunological staining. So you can, this is in the hippocampus, and you can look at kind of the structure of the inhibitory network. In this particular case, the observable is the fraction of PV buttons. So this is a specific kind of inhibitory synapse. And you can see that depending on different conditions, so this is the control condition, this is a condition which is enrichment of the environment, and this is a standard field conditioning. And you can see that the inhibitory subnetwork reorganized significantly, and so there is a change in the structure of the inhibition in the network as a result of this. So how we are to kind of try to understand this complex phenomenology, there are essentially two kind of approaches. I would say they are not alternative, they are rather complementary. One approach is maybe kind of a bottom-up or microscopic, and the idea is take these synaptic dynamics that you have extracted from the experiment, plug it into a model network, and see what happens. This is what has been done with this kind of data, and out of this work has come a very I would say, influential hypothesis about the role of inhibitory plasticity, which is this homeostatic hypothesis that's been developed in these two works, Tim Vogels and Maus Shamir, essentially at the same time. And the basic idea is that as a result of learning, you will have modification in the excitatory connectivity to the neurons, and then the idea is this modification would essentially result in an increased level of excitatory drive to the cell. And then if you want to preserve the dynamic range of the operation of the range, then you have also to increase the inhibitory drive to rebalance the net input to the cell. Hence the name homeostatic. And what they showed is that in fact this specific plasticity rule would in fact result in this kind of homeostatic adjustment. Another approach, which, as I said, I think of it as a complementary approach, is try to bypass all this complexity. As we have seen, single synapse, it's not really a microscopic element in any meaningful sense, because synapses by themselves, they are very complicated dynamical systems. So it's not obvious that you can kind of understand precisely what you're doing. So the idea is take some kind of general framework that allows you to relate some properties of the synaptic structure to some kind of functional level of description. And for what concerns synaptic plasticity, we have this framework. This is a very general framework, which is central in the study of synaptic plasticity in reference to memory. And this is the so-called Hebbian framework from Donald Hebb, who was actually a Canadian, by the way, who actually made this very influential proposal that synaptic modification could, in fact, represent a mechanism for a cellular mechanism for long-term storage of the memories. And this is essentially how these work. So the basic idea is that you have your neural circuits, maybe something in the cortex, and then some stimulus comes in and induce some pattern of activity in these circuits. And this essentially means that some neurons will increase their firing rate as a result of the stimulus presentation, and some other neurons will instead decrease their firing rate. This is something which is routinely observed. And what you see here is triangle are supposed to be excitatory neurons and circles are inhibitory neurons. And darker color means that, for instance, the activity is increased as compared to baseline, and lighter color that is decreased. And so the Hebb proposal is something like this, and it says, okay, if a pair of neurons are coactive during this stimulus presentation, then the synapse connecting them will basically increase its efficacy, and vice versa. If presynaptic neuron, for instance, is highly active, while the postsynaptic one is firing at low rate or not firing at all, then you should decrease the synaptic, the efficacy of the corresponding synapse. So what's the end result of all this process is that you see that now next time that you will present the same stimulus, essentially the input that an active neuron will receive will be increased. Okay, so essentially what you have done, you have kind of induced some kind of positive feedback. Right now, the input received by an active neuron will be increased by means of these kind of synaptic modifications. Okay, so now if you think of the theory in these terms, in the sense that synaptic modification are going to build positive feedback loops, then it's immediately clear that changing excitatory to excitatory synapse is not the only way to do it. There are other ways you could do it in principle. And one is used inhibitory to inhibitory synapse. And here you can see that if you want to have a positive feedback loop, now what you have to do is something that here it's written anti-Hebbian, will be clear in a second. Why? Is because now what you have to do is that if you have two neurons that are strongly active, inhibitory neurons that are strongly active during the presentation of the stimulus, then you have to decrease the synaptic efficacy. So in this sense, this is opposed to the Hebbian prescription for excitatory to excitatory. And vice versa, if one neuron is active and the other one is less active, then you should strengthen the corresponding synapse. Okay, and you can see immediately that this achieves essentially the same result in the sense that it will build a positive feedback loop in the active neurons. And actually, there is a third way, in fact, that you could do, and is to change both excitatory to inhibitory synapse in Hebbian way, so coactive gets strengthened, and inhibitory to excitatory synapse in an anti-Hebbian way. That means that coactive gets weakened. And obviously, you could have, in fact, all these kind of modification at the same time, okay? So now the question is, does it work? And what do we mean by does it work? It means, so this is kind, you see, it's a kind of, at very best, a qualitative theory. So does it work means that now if you plug these dynamics into a reasonable or sufficiently constrained model of cortical circuit, does in fact this kind of modification will result in memory maintenance, and if so, there is something else that we can get out of it, especially in terms of possibly experimental prediction. So next step, I will show how we built this biological plausible or biologically constrained circuit model. So this is somehow, I would say, a standard kind of model circuit. So you have a bunch of excitatory and inhibitory neurons. They are in the cortical proportions, let's say so. So 80% will be excitatory, 20% will be inhibitory. They are randomly connected with some probabilities, and the efficacies have some distribution. What is a little bit more specific to this model network, hence the name biologically constrained, is that all these parameters, so probability of connection, distribution of synaptic weights, are actually taken from experimental measurements in this paper here, where you can see that you have probability of connection between different types of neurons and information about the distribution. And in the same way, we took single cell parameters again from experimental estimates. And so the only free parameters in this model are external inputs that excitatory and inhibitory neurons receive from outside the network. And these were fixed so as to achieve average firing rates as observed in vivo. So this would roughly be about 1 Hertz for excitatory neurons and between 5 and 10 Hertz for inhibitory neurons, okay? So before actually storing memory in this circuit, let me show how the activity looks like. And this is an example of, let's say, intracellular recordings from excitatory cell and inhibitory cell. So blue will always be excitatory and red will always be inhibitory. So you can see that there are significant fluctuation in the membrane, and occasionally you have spikes. Frequency of spike is different because frequency of spikes is different by construction in the model, but otherwise they mimic this Poisson-like nature of spiking process in vivo in the cortex. And another feature of the model that comes because of this random connectivity is that now if you look at the distribution of firing rates into the excitatory and inhibitory network, you will find that these distributions are very, well, they are right skewed. Obviously, firing rate cannot be negative, and long-tailed, and they are very well described by a log normal distribution. So you can see in actual firing rate, and there is a distribution in the log. And you see that this is very well approximated by Gaussian. So in fact, they are log normal. So all of this to say that with this setting, so parameters estimated from experiment and just adjusting the average level of activity in the network, we get irregularity and heterogeneity of firing rates that mimic in some quantitative detail, mimic the activity that people observe in the cortex. Okay, so now we can, sorry, I'm checking the time. So now we can try and store the memories. And how we do this? So I will kind of try to go slowly on this. I don't know if this is kind of, to some extent, is standard memory models. So essentially, first step, you decide how the memories are done. And what we do is that for each neuron in each population can be excitatory and inhibitory, we decide if this specific neurons is to be active in a specific memory. Okay, so this CIA can be 0 and 1, 0 being not active, 1 being active. I means the neuron number, A, the neuron population, so excitation or inhibition, and this mu is just the memory. Okay, so you will have a bunch of binary vectors that will tell you which neuron should be active in which memory. Okay, and the probability of be active is the same for excitation and inhibition, and we took it to be 0.1. Okay, so each neuron will be active with probability 0.1 for each memory. And this probability sometimes is called the coding level, and 0.1 is some reasonable number for coding level in the cortex. Now we have to establish a quantitative criterion for the memory being retrieved or not. And this again, we do following what people do in the corresponding experiments. The idea is very simple. So you compare in a specific state of activity, you compare the average firing rate in the memory population, which means the neurons that are supposed to be active. You compare this firing rate with the firing rate in the rest of the network. Well, in all the network, this is a kind of baseline. And so if this firing rate is higher than the average firing rate in the network, then you say that the memory is being retrieved, and otherwise the network failed to retrieve the memory. Again, I want to stress this is kind of comes out from the theory in a quite natural way, but it's essentially the way in which experimenter decides if a specific neuron is coding for a memory in a real experiment. And now we come to this, obviously the random network will not store any memory, so we have to structure the network, and that is to decide how the neurons are connected depending on the memories they belong. And this we do, again, in a somehow standard way, but with some twist that I will try to explain. So the basic idea that comes from theory is that synaptic efficacy should essentially encode the level of covariance between pre and post-synaptic activity. Okay, so what you see, this is just the covariance of the activity between neuron I and J in population A and B. Okay, so this will be, on average, will be 0, and then if the neurons tend to be coactive, this zeta will be positive, and if they tend to be not active at the same time, this would be negative. Okay, this is just the covariance. Now, this cannot be the synaptic efficacy per se, because synaptic efficacy, so synapses, they come and they are either positive or negative, in the sense that either they are excitatory or inhibitory, but they cannot change their nature as a result of learning. So if it's positive, if it's excitatory, if the synapse starts excitatory, then cannot become inhibitory as a result of the learning process. And to take care of these specific observed fact, essentially we say that the synaptic efficacy is some function of these covariance. And there are different ways to choose this function. I will discuss maybe alternative ways at the end, but obviously one constraint is that this function is always, in the case of excitatory synapse, is always non-negative, and in the case of inhibitory synapse, is always non-negative. And then Hebbian or anti-Hebbian nature of the synaptic structure depends on. So this will be in the case of excitatory synapse, this will be a growing function of Z, and vice versa. In the anti-Hebbian case, this will be a growing function of minus Z. Just maybe this is clear if I just give an example here. So this is the Hebbian term. This will be this covariance. And as you can see, this can be positive and negative. In fact, this will be on average 0. And this is, for instance, a function that implements E to E plasticity. So you can see that if this is negative, basically the synaptic efficacy is 0. And then at some point, it starts growing. And this choice is motivated by trying to match actually this function is chosen as to match the marginal statistics of excitatory to excitatory synapse measured in the experiment. And this is the same for the inhibitory to inhibitory synapse. And you can see that, in fact, in this case, if this is positive, actually the synapse is 0 and then starts growing as a function of minus Z. Okay. I think I've said everything. I don't get any feedback, so it's complicated for me to understand if you're following. Okay.
Yes, we are following. And just I take the occasion, you still have about 10 minutes. Okay.
Perfect. Thanks. Okay, that's it. So let's see now finally if this in fact works. And this is an example from the network I have just discussed before. And storing, in this specific case, we store 2,000 memories in the synaptic structure in the way I've just described. And here we probe if the network is indeed able to retrieve the memories. And how this works, so these gray bars that you see, this is mimicking external simulation. So what we do is just we inject transiently some external inputs to the neurons that belong to a specific memory, so as to activate it. And then when the gray bar is finished, this input is being removed. And you can see that, in fact, the network is able to retrieve the memory. And you can see that these are spike clusters. So you see that the spatial distribution of activity initiated by the stimulus presentation is, in fact, maintained automatically by the network when the stimulus is removed. And now here different stimulus comes in. You see that the spatial distribution of activity changes as a result of the stimulus presentation. But then again, this is preserved and so on and so forth. Okay, this is for 3 different memories out of these 2,000. And this is a slightly more precise measure of the retrieval quality, and this corresponds to, it's called the overlap. This just corresponds to the correlation between the actual firing rates in the network and this binary vector of 0 and 1 that are supposed to represent the binary coding of the memory. Okay, so positive overlap, that means that the state of activity of the network is correlated with this vector, which means that active neurons, so neurons with the 1 are firing at higher rate than neurons with a 0. And you can see, again, the different colors are for the different memories. So you can see this is, let's say, the black memories is being retrieved, so the overlap is positive, and the other have overlap at 0. Here, different stimulus comes in, black goes down, this green goes up, and so on and so forth, okay? So it seems that this idea can work in the sense that in a network that reproduce some features of the cortex, this mechanism seems to be able to function. And now that you have such model, one of the values of doing this kind of modeling work is that now you can ask questions that are more complicated to ask in an actual experiment. And one question you might ask is, well, at any rate, is the question that we asked is, which one of these mechanisms, in the sense of this kind of different form of plasticity, are more important for storing the memory? They have the same efficacy or not? One simple way to do this is to do what we called rewiring, which I'm going to explain in a second. So here, the fact that you don't see this exciter-to exciter connection, that means that this has been rewired. What does it mean, rewired? It means that essentially we generate the E2E connectivity exactly with the same marginal statistics as before, but there is no correlation with the memories being stored, okay? So everything is the same, marginally speaking, but in the E2E connectivity, there is no information about the memory. Obviously, the information about the memory is preserved in the other connectivity, and that's why you see that even after the rewiring of the E2E connectivity, the memory retrieval of the network is intact. You see that there is very little change in the retrieval dynamics. And now you can keep and play the same game. And so one thing that you can do is that you can rewire eye-to-eye connectivity, which is indicated by the fact that now you don't have this red arrow here. So synapses are there, but they have no correlation with the memories being stored. And in this case, you can see that now the, retrieval memory performance of the network has been affected. In fact, you see that essentially the retrieval state disappears, okay? So basically this simple experiment suggests that in fact synaptic structuring in the E2E population is more effective or more important in storing memory than the synaptic structure in the E2E connections. So now this is, or should be, kind of surprising. And one of the reasons this should be surprising is that, in fact, actually, there are the number of E2E synapses much, much, much larger than the number of eye-to-eye synapses. So, in fact, it seems that even if they are small in number, absolute number, they are more important. Also, I would like to stress that standard idea or consensus about the let's say the locus of storage of the memories, most people think that this is in the E2E connectivity for several reasons. And these results suggest that, in fact, this seems to be I will elaborate in a moment, seems to be the less effective place to store memories, at least under certain assumption that I will shortly discuss at the end. Another advantage of having a reasonably simple model is that part running this kind of fake experiments is that maybe you can do some theory and compute some quantity of interest in this simplified setting. And obviously one quantity of interest in this specific case is how many memory I can store depending on where I put information in the synapse. Okay, so let me illustrate this. So what you see here is measure of storage capacity. So this is the number of memories you can store per neuron. And what you see on the x-axis is where you decide to store the memory. So this means that you decide to store the memory only in the E2E connection. Here you decide to store only in the I to I connection. Here you decide to store in the I to E and the E to I, and so on and so forth. Okay, and this is the simulation we started with, where information is stored in all synaptic population. Okay, so first striking fact is that, as you can see, and I was anticipating before, actually E to E connection seems to be the least effective for storing memories. And already just using I to I, you can do basically 4 times better. And now if you consider the absolute number of synapses, it turns out that here you're doing almost 25 times better per synapse. And then there are different ways, and it's interesting that even storing the excitatory to inhibitory and inhibitory to excitatory, actually you do better than E to E. And clearly, if you store everywhere, you do better. This is the 5 minutes?
Yes, I have to ask you to wrap up because...
Okay, no, I'm finished. I still have 5 minutes.
Thanks.
Or no?
No, well, you should have less, but I allow you 5 minutes. So
no, okay. So I'm going fast. Okay. So
thanks.
As I was saying, we can understand to some extent this behavior in the model, and I will not go into detail because I don't have time. So let me just go to the bottom line. So the bottom line is that the signal that you get in the maximal signal that you can get in such a network that operates in a balanced regime depends on the average firing rate of the corresponding population. Okay, so essentially the advantage of inhibition over excitation in these specific settings, so in a balanced network, depends simply on the fact that average firing rate in the inhibitory population is essentially 5 to 10 times larger than average firing rate in the excitatory population. And I'm finished. What I've shown is that essentially extending this Hebbian framework to include inhibition. First you find that inhibition is perfectly viable to store inhibitory synapses are perfectly viable to store memories as excitatory synapse. And actually, it turns out that if the network is operating in the balanced regime, it turns out that, in fact, storing memories in inhibitory synapse is actually a better solution in the sense that you can store much more memory per synapse. This also suggests a theoretical study that suggests that previously ignored sites of synaptic storage, in fact, like from inhibition to excitation, in fact, seems to be very effective, at least in the context of this simple model. And finally, there is also kind of experimental prediction that in fact says that if you want to store memories in inhibitory synapse, then in physiologically relevant condition, plasticity at inhibitory synapse should be anti-Hebbian rather than Hebbian. And I will stop here. These are just maybe I can address in questions, or it's okay. I'm done. Thanks.
Thank you.
Thank you, Gianluigi. And as we are running a little bit late, I would like to ask the audience to keep on asking questions in the box below. And we are moving the questions that you have for all of us during this session to the first part of the discussion panel at the end. So the speakers will kindly agree. I mean, they haven't done yet,
Sure.
but they are doing it right after this statement to be there later. Okay, so thanks,
Okay.
Gianluigi, again, and then
Welcome.
we can move
Thank
to the
you.
next speaker. Thank you.